And continuing on a little bit with what Doug had talked about earlier, one of our commitments is definitely to drug delivery at AbbVie. But I would like to draw your attention to just what came out. It's ahead of print on the EPUB from the Glaucoma ophthalmology. And it's an editorial where, and AbbVie did help contribute the group getting together to help discuss this, but we felt that it was really important to take a look at the struggle that patients do have with adherence to drops. And through this group, they evaluated the different challenges that patients have and said it's important for us to try to identify these people and consider what resources are available to help them address the problems that they have with adherence, and try to tailor your strategies towards how you address the patient care for those people that do have problems with adhering to their drop regimens. Now, AbbVie is committed to helping address non-adherence and through drug delivery. Our first foray into this started over 12 years ago and was approved just about 5 years ago, and that was Durysta. And it, as you know, is approved for a single administration. And how the trial design initially was set up was every 4 months there was fixed administrations because we felt as the drug was eluting, there would be a wearing off, we thought, of the effects of the drug. And what came out of the studies, we did find that there was some corneal endothelial cell loss that was associated with the repeat administrations. So we pivoted to looking at a different way of approaching what trials we had already designed at that point to continue to evaluate the Durysta implant. And what we did then is we had in place 2 studies that were ongoing. One was the Athena trial, which is basically 180 eyes, and it was a paired eye trial that were receiving 10 microgram bimatoprost implant in one eye and a 360 SLT in the other. And there was repeat administration allowed in this originally before the FDA approval. So we had to pivot and change from a fixed regimen to kind of a flexible. We thought that'd be more appropriate, given some more time between the implants. And then the Triton study was a PRN dosing study where repeat administration was allowed in that trial, but time could go on for longer than we had with the Athena trial. And then finally, the Argos trial is basically a phase 4 real-world study, which just evaluates what happens with a single administration alone. So with the Athena, basically what we found from that, and it's available online, and the references on the bottom there, but what we found is that the safety, which was one of the concerns that we had with the implant, with 2 administrations was if it was on a fixed regimen where it was every 4 months somebody got it, the endothelial cell loss was still in the 5 to 6% range. And what we found is when we went flexible, it dropped down a little bit closer to the 5% range. SLT had endothelial cell loss at about the 3%. And this is using a 15% cutoff for the endothelial cells. But what we really wanted to try to focus on, and that's what I wanted to get to here today, was really what did we find when we look at a single administration alone where time is allowed to go on between the administrations. And this was a Triton study, which is the PRN dosing, and you'll see the number of eyes at risk on the bottom there. But as you take a look at the survival curve, at a year out, after subjects are allowed to go in towards a PRN type of dosing, and there was no criteria set for what determination the doctor had to use to re-administer, we see over 50% of the eyes made it out a year with survivability through the Triton trial. We did see safety-wise that there was a slight drop in the overall endothelial cell count over a period of time. As far as the Argos study, which is what I really wanted to just focus on for a minute here, we tried to get a little bit more specific towards what was applicable to a single administration being used in the population. So we set a target IOP. So when doctors did decide to retreat, there was a reason for it based on the IOP. We did set an endothelial cell criteria, but basically what the Argos study was a real-world study allowing patients to come in, get the Durysta implant, and then be observed over a period of time to see what happened. As you'll see, the most common reason that people were using it was intolerance to drops or noncompliance that was feared to be present for the patient. Limitation of the study, obviously it's a real-world study, so we did lose some patients over a period of time with it. Primary endpoint was to take a look at the number of eyes that made it out to 6 months without needing some kind of a rescue. Now, patients came in on a certain number of drops with this trial. They were not washed out. And then they were administered the bimatoprost implant and then followed from there on out, and medicines could be reinstituted if they felt they needed to based on the target. So here's the primary findings out of this. What you'll see is the mean for the population was at about 17 initially. We had set a target there. The target had been determined for population at 15. And after the implant was administered, the IOP dropped down to the mean that was desired for the target at 15. And the number of drops dropped from baseline at 1.8 down to under a single medication through the 18 months out to the trial. As far as TAEs, corneal endothelial cell loss was at the 1.5% that it was reported. And the cell counts remained relatively stable and similar between both the auto count and the reading center. So I'd like to thank you. All right.
Good afternoon. I want to thank the Glaucoma Foundation for giving us an opportunity to make this presentation. So Igenix has a three-pronged approach to combat irreversible blindness due to glaucoma. As you all know, in fact, the previous speakers talked about beyond IOP. I think the major issue in terms of glaucoma is the death of retinal ganglion cells. There's about 10 to 15% of all the glaucoma patients lose vision due to eventually lose vision, even though they have optimal IOP. So what's our solution? Professor Ram Nagaraj at the University of Colorado Department of Ophthalmology has been working with small heat shock proteins that are anti-apoptotic proteins, and he has shown that there is a significant prevention of cell death, the retinal ganglion cell death, upon insults. He has published a number of papers. Igenix has licensed the patents from the University of Colorado, and we have been working on some of these assets we licensed from the University of Colorado. We have three assets and one principal target, which is the heat shock protein, small heat shock proteins. And the first one is a peptide, which is about 20 amino acid peptide, which is in the core part of this protein. And we are focusing that to acute indications like the acute angle closure glaucoma and NION. And we have the second one, the gene. He has developed expressed the gene with a retinal ganglion cell specific promoter and delivered it with the AAV2. He has done a lot of work in animals, and we recently have done some work in the non-human primates. So that we are focusing on the chronic indications, such as primary open-angle glaucoma and normal tension glaucoma. The last one is gene-engineered stem cells. It's kind of very early, but it's very interesting in the sense he has been working recently on taking human iPSCs, growing them, making retinoids, and isolating pure RGC cells, and then transfecting them with our gene for heat shock proteins. And in the early studies, he has shown that in optic nerve crush model, after about 2 weeks, there is a significant number of these human retinal ganglion cells engineered with our protein were able to establish, and not only structurally, but also functionally. Of course, it's very early. He hasn't done this axonal growth into the cortex and all that, but initially the functional assays does indicate there could be a potential. I mean, a lot of work needs to be done, very early stage. So the peptide, I'm not presenting all this data because we don't have much time, but I'll be happy to talk afterwards. But there are some articles that I have quoted here. The peptide, like I said, is a 20 amino acid peptide of these small heat shock proteins that significantly reduces the death of the retinal ganglion cells in a number of models, almost like 5 models he looked at in rodents, and also in human cadaver explants. If you leave them for about 7 days in the media, about 50% of them die, but in the presence of the peptide in the medium, there is a significant reduction of the death. Again, it's all published, and you're welcome to I'd be happy to provide that information afterwards. And the gene data, again, like I said, AAV2 delivered gene with a specific promoter that only expresses in RGC cells. And what is interesting is this is a published paper, but there is an unpublished data where we gave one injection of this AAV2 in glaucoma mice, I think microbead model. And then after a year, if you look at the animals, if you look for the protein in this retina, and there is a significant production after 1 year of the injection. This is almost like a 30% of human life. If we extrapolate in mice, they live about 2 years, lab mice. And there's also improvement in the vision, compared to the glaucoma mice. And lastly, we've done 2 non-human primate studies. One of them is to look at initially the gene expression and the dose response. And there was an excellent transduction of the gene in all parts of retina, both in soma and in the axons. And then we completed recently an efficacy study where we saw about 30% improvement in vision. This is in NHPs, obviously. I just want to show you. Here, you can see there is, so we looked at the dose response initially, and you can see in fovea significant expression of the protein, not only in the soma, but also in the axons. And again, in this picture, showing it's not only in fovea, throughout the retina, we see this. Obviously, there is a dose response. At high doses, we can see significant expression. So what is our business model? We plan to take all 3 assets forward into clinical trials towards FDA approval. Obviously, we cannot do on our own. So hopefully, we're looking, and hopefully we'll find some partnerships for funding from investors and strategics. The team, like I said, Ram Nagaraj is the primary scientist, and we have Meena Panchuteva. She's also a glaucoma expert at the University of Colorado. And we have a team we think we can take up to clinical trials and hopefully can pass on to strategics later on. So again, as a summary, we have 3-pronged approach to save vision from glaucoma: peptides, acute glaucoma, and NIAN, gene therapy, chronic glaucoma, normal tension glaucoma, and then engineered stem cells, hopefully for restoring vision. Like I said, it's too early, but we're really excited about it. Right. Thank you very much for your time.
Good afternoon. I would like to thank Courtney and Paul for the opportunity to speak, and I'd like to congratulate Adrian and Andy for putting together another fantastic Glaucoma 360 meeting. So it's been an exciting close of the year at Glaukos in 24, and it's been even more exciting start of 25. And so within this context, it is my privilege to share with you an update on the progress achieved within the R&D pipeline, with special focus on our glaucoma products in development. And before I proceed, I would be remiss not to acknowledge the terrific contributions from the clinical investigators and colleagues from the US FDA, without whom this progress would not be possible. So these are our forward-looking statements. As you know, more than 20 years ago, Glaukos was founded to transform glaucoma care, which led to the iStent mixed product. But we've continued to innovate, and over the last 6 years, we have invested over $700 million into R&D. So a large portion of this R&D investment went into our glaucoma pipeline, which is based on a balanced mix of pharmaceuticals, medical devices, drug-device combinations, and diagnostics. Today, we would like to share with you 3 new data sets: 3-year evaluation of iDose TR based on 2 pivotal studies, robust efficacy of iDose TR in combination with cataract procedure, and also head-to-head comparison of iStent Infinite with HydroStent based on our integrated head-to-head study in mild and moderate glaucoma patients with standalone administration. So starting with iDose, iDose is a microscopic implant that resides at the iridocorneal angle. and delivers multiple years of travel-post pharmacotherapy to reduce IOP. iDose was approved as a pharmaceutical, and from that perspective, it has a very broad labeling covering all disease severities. I would like to direct your attention to the scleral anchor feature of the device, which immobilizes iDose at the iridocorneal angle and results in an excellent corneal endothelial cell safety. So we would like to share with you today 3-year evaluation of long-term safety and efficacy based on completed phase 3 pivotal studies of iDose. And we are pleased to report that these phase 3 studies confirm long duration, with 70% of iDose patients remaining well controlled for their IOP after a single dose at 3 years. Similarly, from the standpoint of long-term safety and efficacy, the tolerability and safety was also excellent, showing absolutely no adverse events of corneal endothelial cell loss, no AEs of periorbital fat atrophy, and very little incidence of conjunctival hyperemia. So these are exciting results that truly replicated the phase 2b study evaluation that also was conducted over the course of 3 years. Second data set I would like to share with you is the robust efficacy of iDose TR in combination with cataract procedure, wherein iDose plus cataract reduced IOP by 11.3 millimeters of mercury, or 44% change from baseline. And so given this robustness of the IOP lowering, one can speculate that iDose TR combined procedures have the ability to replace perhaps 2 or perhaps even 3 topical IOP lowering medications. From the standpoint of combination procedures, we are also evaluating the efficacy of iDose TR in combination with iStent Infinite. As you know, iStent Infinite was cleared by the US FDA for standalone administration in refractory glaucoma patients. So this is work in progress. The third and final data set I'd like to share with you is the head-to-head comparison of iStent Infinite with HydroStent, conducted in mild and moderate glaucoma patients via standalone administration. In this head-to-head comparison, iStent Infinite demonstrated statistically significant and clinically relevant superiority in IOP lowering in unmedicated mean diurnal IOP, conducted as a change from baseline as well as percentage of responders in eyes that had no surgical complications. So based on this robust efficacy that we've seen represented by iDose TR using drug delivery approach or iStent Infinite using the mix approach, we can state that it is truly time to change. It is truly time to change the traditional paradigm of topical eye drops first and replace it with SLTO drug delivery first, followed by MIGS, to really truly deliver the best care of our glaucoma patients and put the control of the disease where it belongs, which is in your hands, in the hands of the clinicians. So in closing, let me touch on 2 pivotal studies we are excited to report on. In March of this year, we'll be starting pivotal study of PreserFlow in the refractory glaucoma patients. And at the close of the year, we have already commenced phase 3 pivotal program of iDose TREX, which is our second generation iDose TR-based product that is aimed to deliver 6 years of pharmacotherapy and IOP lowering after a single dose. So given this exciting progress in the Glaukos pipeline, which is truly synergistic with the progress achieved with our cornea, rare disease, anterior segment pharmaceuticals, and retina pipelines, we can truly state that we are executing well on our R&D strategy and transforming and improving lives of ophthalmic patients worldwide, while giving them the most valuable gift which we can give, which is the gift of sight. Thank you so much for your attention.
Commercializing drug delivery from contact lens glaucoma. So today there are 2 approaches to treat glaucoma, either eye drops or alternate approach like implants or surgery. We fit in the middle of these 2. We believe at MediPrint, we are providing an optimal solution where we have manufactured a contact lens that will deliver bimatoprost continuously for 1 week. It's an optimal solution because it meets the need of patient, practitioner, and providers. So the idea of delivering drug from contact lens is a very old one. In 1965, it was patented. And since then, significant research has been done, yet there is no commercialization, because what they did was put the drug inside the lens by soaking it or by creating a de novo lens. When you soak it, the drug comes out very fast. When you make a de novo lens, it requires new manufacturing process or new drug or new lens. FDA is risky. So investors are staying away from it. So what we have done at MediPrint is leverage the digital printing technology and made it a platform technology. What we do is print FDA-approved drug on a surface of a contact lens. And also we, like a multicolor inkjet printer, put multiple drugs or materials in the inkjet printer cartridge and allow it, print it multiple times to create a structure that modulates the drug release rate. So, and what happens is when we inkjet print, then rest of the process is existing manufacturing process. So what are the advantage of it? We have added only one step to manufacturing process is inkjet printing. So we achieved the scalability as well as the seamless integration with existing manufacturing. Of course, we got a regulatory ease because we are using FDA-approved drug, FDA-approved contact lens that gave us a 5 to 5 pass. Also, this novel approach required innovative solution. As a result, today we have more than 28 patents in our global portfolio. How did our lens perform? We recently finished the phase 2B dose escalation study. During this study, we found our second dose of 32 microgram gave us a 30% reduction, which was equivalent to 0.01% bimatoprost solution. Also, you will notice that the frequency of the treatment required, what contact lens was required to insert only 6 times compared to 42 installation of bimatoprost. If it was timolol, it would be 84 installation. And since our product is used only one time and is sterilized, we do not need preservative. And we address the issue of discomfort with contact lens by simultaneously delivering HA for 7 days as an excipient. And we found that it to improve comfort as well as dryness by about 40%. So overall, if you look at it clinically, our lens was found to be safe because there was not a single dropout. Even the percentage population was from 73 to 84 in one trial. Also, we found it to be safe. There were no treatment-related adverse event. Having proven the clinical validation, we wanted to address the adoption. Today, 140 million people wear contact lens. It's a proven carrier. Our product is comfortable, is at low cost, and non-invasive. And as I discussed before, it's very convenient. Somebody who needs assistance in putting eye drops or contact lens, the number of time person will have to visit will go significantly down. And also one thing to keep in mind that we are not changing the glasses for a contact lens. We are treating and preventing blindness. So our product meets the need of patient because it's comfortable, convenient, low cost, and it's good outcome. The practitioners are also happy. Not only they get better outcome, but now they get to dispense the contact lens, so they will have some economic benefits. And manufacturers have low capital investment, low cost and high margin product. and it's a platform technology. There's many applications after glaucoma. It could be dry eye, it could be post-cataract surgery, so there are applications to many more products. So what have we done? In 2014 at OIS, they gave the desirable attributes of product for ocular drug delivery. And that included high compliance, the non-invasive nature, particularly of easy removal, no debris. So we meet all those required. And today, we will truly have a platform that is transformative. I think it's going to transform the delivery of ocular drug as well as add to the functionality of contact lens. Keep in mind, it's also life cycle management tool. It's applicable to existing product or new product. So what are we going to do with it next? This year, we are addressing end-of-the-day comfort issue for a contact lens wearer. Those who are here know contact lens wearer know it feels dry and itchy. With our HA, we found it to be very comfortable and reduce dryness. And that's going to be 510(k) approval. Following that, we are going to continue our phase 3 march towards the approval of our lens with the BIMAT approach delivery. Following that, we have a HA that is approved for dry eye in Europe and Japan. We'll pursue that. So we have a product pipeline that we are going to have a market reach about 11 billion today. What we ask is, so far we have invested raised 7.2 million. We are looking for a bridge loan of 7 million, and long-term our need is $35 million. I thank you, Glaucoma Society, for this opportunity, and I invite investor community to join our journey for a paradigm shift in glaucoma treatment. Thank you very much.
Spinal cord and CNS do not regenerate. This is our problem with glaucoma. However, have a look at those rats in which the spinal cord was cut. Look at the left one and the right one. Of course, the left one is the control, and the right one was treated. So we are now looking into going into clinical trials with the spinal cord regeneration, but this is very complicated. It is almost impossible. So we decided to go into glaucoma, which is much more common and much simpler, but still we have to show you these pictures from the spinal cord. You see the regeneration in green, and it seems that we made a first in medicine, and how it's done is doing siRNA to prevent TPN, which is the molecule that prevents regeneration in the CNS, and delivering it by exosomes, which seems to offer very good delivery system which can deliver the siRNA right to the right place of inflammation. Actually, in the spinal cord trial which you show, we delivered the exosome with the siRNA to P10, what we call exo-P10, intranasally. We didn't have to bother even injecting into the spinal cord. And we don't know yet we didn't do a dose response yet, or maybe injecting it intrathecally will be better. However, we decided to go into ophthalmology, and I thought I should have showed you, this is the system. how it works. The cRNA for against P10 is delivered, and it seems to be doing the work. I don't have time to go into the details. And I did an optic nerve crash, which is a good model initial model for glaucoma, optic nerve crash, and injected the exopitan. This time we didn't go it intranasally, we injected retrobulbarly. And after 6 days, we did an OCT and ERG. And again, after 17 days and 19 days, we did ERG again and OCT again. Of course, in ophthalmology, we are much better off than in the spinal cord that we have numerical data immediately, almost immediately. And as you can see, the spinal cord, the ERG recovered in the injected animal, even better than in a control animal, and even in the animals without optic nerve crash at all. I don't know how it worked, but it worked. And what this is noticed in the right-hand form, the optic nerve crash and exopitan had a better ERG than the eye which wasn't crushed. So this is the data that we have. We only started the project with the eyes about 2 months ago. And this is all the data we have now. But, oh, sorry, I should have told you we did measure the number of ganglion cells in a flat mount. And as you see in the left hand, the control, the exopitan on the right hand, and with exosomes, without the exopitan, there is no improvement at all. And those are the number on the graph. Looks okay to me, and it's certainly worthwhile continuing this project. And this is the program. We'll do more optic nerve crash trials. We don't have enough animals even for an article. And then we'll do an acute glaucoma model with a rabbit laser and try it then another 6 months. And then we'll do a human trial within a year on acute glaucoma, because we had the model of acute glaucoma. And of course, if it works, it will be nice. Also, I plan to do facial nerve palsy. I don't know whether the system will work on peripheral nerves. I don't know. But the model is so easy, and the clinical trial is so easy, so it's worthwhile trying. And then we'll do an RCT of the two trials of above, and we go through all the bureaucracy. And then I would like, my program is to do open-angle glaucoma, severe optic angle glaucoma. Maybe we can regenerate or prevent from dying of some of the nerves. As you know from Quigley's works, in every stage of glaucoma, there are about 30% of the fibers in the optic nerve in the process of dying. Maybe we could save them. Again, the trial will be simple, and we are now going through, the part of the spinal cord people are going through the good manufacturing practice and the regulatory and the acute toxicity. And by the time we finish, they'll have their data, so we can go straight ahead to human trial, I hope. And as for financing, I invested about $350,000 for my Alcon money, and because this is a very good investment. And anybody wants to join me, please queue up. Thank you.
Hi, everyone. I'm Therain Tu, the CEO and co-founder of Qlaris Bio. On behalf of my co-founder, Dr. Barbara Barasco, our investors, and the entire Qlaris team, I'd like to thank the organizers, and especially Dr. Adrian Graves, Dr. Andrew Ewok, for once again inviting me to provide an update on our progress at Qlaris. As we all know, approved drugs targeting intraocular pressure currently target only 3 out of the 4 components of intraocular pressure: aqueous humor formation, uveoscleral outflow, and conventional outflow facility. None primarily target the 4th factor, episcleral venous pressure. As such, EVP continues to set the floor for IOP lowering and in preventing optimal IOP control in patients with glaucoma. At Qlaris, our mission has always remained singular. That is, to lower that floor by targeting primarily episcleral venous pressure. QLS 111 is a first-in-class, preservative-free, ATP-sensitive potassium channel opener formulation that serves to relax vascular and vascular-like tissues of the eyes, to primarily target EVP, and to improve drainage beyond the trabecular meshwork. What we think we are providing now with QLS 111 is a product profile that delivers not only superior additive IOP lowering, but in a way that actually brings unparalleled safety and tolerability, underscored by a long history of not only ocular administration, but systemic administration as well. As such, QLS 111 may serve as an ideal complement to all available drugs and drainage devices in patients with primary open-angle glaucoma and ocular hypertension. Importantly, it may also serve as the first potential drug to be indicated for the lowering of intraocular pressure in patients with normal tension glaucoma, where EVP happens to play a disproportionately large role as a component of total IOP. Today, I'd like to give an update on our phase 2 development program around 2 studies that we have just recently completed that were both masked, randomized, and vehicle-controlled studies in patients with primary open-angle glaucoma and ocular hypertension. These were the Osprey and the Apteryx studies, and both studies had actually completed having met all primary and secondary endpoints. Shortly, we shall also be enrolling the Nightingale study, which will be studying QLS 111 in patients with normal tension glaucoma. In terms of a little bit of detail around these 2 first studies, the Osprey and the Apteryx studies, in the Osprey study, we were using QLS 111 as monotherapy to determine not only safety and tolerability, but also an optimal dose and regimen. In the Apteryx study, we used QLS 111 on top of latanoprost in patients who are stable on latanoprost monotherapy. In the Osprey study, when QLS 111 was used as monotherapy, the 0.015% dose, when dosed as a once daily in the evening dose, lowered mean diurnal IOP by 3.7 millimeters of mercury from a baseline of about 23. In the Apteryx study, QLS 111, 0.015%, when dosed on top of latanoprost monotherapy, lowered mean diurnal IOP by an additional 3.2 and 3.6 millimeters of mercury when dosed as either QPM or BID dosing. Importantly, in both studies, there were no clinically relevant ocular or systemic adverse events, no hyperemia when dosed as a monotherapy, and no added hyperemia when dosed on top of latanoprost monotherapy. This level of IOP lowering, we believe, surpasses the available combination therapies on the market today. And this adverse event profile is certainly one that we are quite pleased with as well. As you might imagine, and as we've heard earlier, compliance and adherence are a major factor in the outcomes of patients taking topical eye drops. So in summary, with QLS 111, we believe that we have an ideal complement to other drugs and drainage devices for better IOP control. Importantly, we are the first and only product that primarily targets episcleral venous pressure with a unique mechanism of action that appears to be almost completely additive to prostaglandin analogs and delivers greater than 3 millimeters of mercury of additive IOP lowering when used with latanoprost. The formulation itself is preservative-free and one that also has an exceptional safety and tolerability profile, again, with no added hyperemia. We're excited to continue developing QLS 111. We're excited as well to be starting the study in normal tension glaucoma shortly. And I'd like to thank again everybody for listening to my talk. Thank you.
Thank you very much, and thank you to the organizers, Adrian and Andrew, for the opportunity to present today. It's a privilege to take you through the SpyGlass Pharma story. SpyGlass started 6 years ago, and we're developing a long-term, multi-year drug delivery platform to address chronic eye conditions at the time of routine cataract surgery. We're starting in glaucoma. We've known for decades the medicines that we have access today to lower intraocular pressure work very well. Unfortunately, they can't work when patients don't take them. We intend to solve that. Our lead product is targeting 3 years of bimatoprost therapy for patients with glaucoma or ocular hypertension. We know the massive unmet need is significant. The SpyGlass approach is unique. We're combining a known drug with a known device and a known procedure in an elegant way that all cataract surgeons already know how to do. We announced last year we completed enrollment of our US phase 1/2 clinical trial, and we look forward to moving into our phase 3 pivotal trials later this year. We have active pipeline development to extend patients beyond 3 years, and we can also elute other products, APIs, for other ocular indications, and we'll talk about that. The SpyGlass system consists of 2 parts. First, a single-piece hydrophobic acrylic IOL. We're starting with a monofocal, but we also have plans for other advanced optics like torics and EDOF lenses. You've noticed from this animation, we've modified the haptic optic junction to receive 2 non-bioerodible drug-eluting pads that securely attach at the haptic optic junction. From this point forward, it's cataract surgery. We're using an off-the-shelf IOL injector. We're implanting through standard cataract incision sizes. The entire system is implanted entirely into the capsular bag. Lens unfolds, similar flat times to commercial IOLs, and the drug pads remain outside of the visual axis, continuously eluting bimatoprost 24/7 for 3 years. The lens stays in place, the drug pads do not change in size, shape, or volume, and once done eluting product, it's essentially part of the intraocular lens. Millions of patients are waiting. 1 in 5 cataract procedures done around the world are done in patients with glaucoma or ocular hypertension, 20% of our procedures. That equates to 2 million annual procedures in the US and Western Europe, and another 4 million procedures around the world. Despite the fact that 1 in 5 cataract procedures are done in patients with glaucoma, only 35% of our cataract surgeons today are routinely deploying MIGS type of technology in their cataract surgeries. That's greater than 1 per month. We can do better than that. With SpyGlass, 100% of cataract surgeons can implant our technology, essentially 3xing or tripling the available surgeon pool currently employing MIGS procedures today. Well, how do we know we can do this for 3 years? Simple. We've already proven it in the laboratory. I'm being coy here, showing you only over 1,000 days, but we know we can go for longer. We can get beyond 3 years, but surgeons told us very clearly, give me something that lasts 6 to 12 months. We said, that's not good enough. We can do more. So we're starting with 3 years. Beyond the laboratory, well, that's nice. We needed to prove this. What happens when you put this in patients? We embarked on a first-in-human feasibility trial a couple of years ago down in Honduras. We enrolled 23 subjects with a visually significant cataract, and they also had glaucoma They were on active medical therapy for IOP-lowering drops between 1 and 3 bottles. We randomized them to one of 3 dose strengths, the SpyGlass IOL, and we continue to follow them up today. These are the 18-month results. 18 months after implantation, the IOP lowering is remarkable. Mean IOP reduction across all groups is 43.7%. All patients were at or below 18 millimeters of mercury, and 100% of patients remain off of all topical IOP-lowering therapy 18 months after implantation. Visual acuity was also remarkable. 100% of patients were 20/30 or better all the way through 18 months. And there were no product-related adverse events. What we see are things we see in standard cataract surgery. A couple of patients with mild dry eye that resolves with use of artificial tears. As we talked about this morning, pathway to approval is important. This is a 505(b)(2) pathway. And from a reimbursement standpoint, we're already actively thinking about this because we've heard so much today about building reimbursement confidence takes time, it takes thoughtful consideration, and after approval, we intend to apply for a J-code, and this will be reimbursed ASP plus 6%. We also see pathways for additional professional fees as well. From a pipeline standpoint, we look forward to moving into our phase 3 pivotal trials with our lead asset. Our secondary asset is essentially a standalone procedure. It's a platform extension implanted anterior to the capsular bag to be able to give the patients years of additional therapy and also address the millions of pseudophakic patients out there that need our help. We can also do this with other molecules, small molecules for potentially AMD treatment, as well as cataract or long-term control of uveitis. The leadership team at SpyGlass is no stranger to ophthalmology. The company was co-founded by Malik Cahook and Glenn Sussman. Malik is here somewhere today, so we'd be happy to answer questions later. I serve as CEO, and James Denwell is our Chief Operating Officer. We have great financing. We have great backers. We have strong support. We've raised over $120 million to date. We're moving through our phase 2 and into our phase 3. And ultimately, our plans are to do that this year and then continually building the road to taking the company public. Thank you very much for your time and attention.